Hi, my name is Vishal. This is Ron and Roman, and we call our platform RAS, or Research Assay Standards. And RAS is a methodology licensing platform for cancer researchers. Irreproducibility is a concept close to cancer researchers' work. Scientific news and editorials are awash in pieces discussing and trying to measure the amount of irreproducible research. In 2012, Amgen claimed to have been unable to reproduce 47 of 53 landmark cancer papers and chose not to publish that data. But in 2016, they published data showing that they were unable to reproduce data from 3 high-profile journals. So this consensus has led the NIH to add 4 new grant criteria to boost reproducibility, and studies, such as the one from Boston University, have claimed up to half of all money spent privately and publicly are being spent on irreproducible research. So our platform, RAS, contains 2 things: a website where researchers can submit their papers in order to confirm that their papers meet a particular methodological minimum standard, and a browser extension where cancer researchers can browse PubMed with annotations for the papers as to whether they do or don't meet this minimum standard. And the outreach to cancer researchers will consist of emphasizing limiting the waste of both materials and labor on experiments which fail to produce reproducible data. And Ron will now show you the platform.
Aspects of our app, which is the web extension. So let's go to Chrome and go to our beloved website, PubMed. Let's say RAS, right? All right. So let's go to this article. Click. Did you see this extra stuff?
So
this is the extension. So we pull in the data right there. See, so this paper, let's say it's submitted for review for us, but it's waiting for results. Okay, 80% completion. Let's go to another article. The second one, for example. See another. This one is being proved, and those methods can be verified, and you can take the paper and do the experiment. Now, so if you want more information, you go to our website. Okay. So we have an institution. We call research assay standard. That's the S. The other meaning of that. So we pull in the scholar information, whatever paper has been certified or under reviewed. Okay. Now you can submit a paper. Let me show how to submit a paper.
Still have the book, so I don't really know what's going on.
All right. Anyway, so it's a URL. So this is a URL. Type in the URL. Submit. All right. And you fill the questionnaire. This is key. So those things, those are the protein, whatever, those keywords in our bag of words, corpus, let's call it EMPL. So you have to type the exact location to verify. So in there, your paper, so paper page number 2, line number 3, fit that criteria. Once you're done that form, you go next, next. So this is like you filling your tax. It's an AI type going through. All right, so then you can submit. All right, we submit, and let's see what happened. Submit. Then let me go to my, by the way, this is the paper. That's the URL. Yeah, that's the paper I put up. All right, I go to my website. I'll go to my email. I say, okay, your question being received. Okay, it's all in real time. And those are the
A lot of researchers will complain that the review process takes so long already, about 1.5 years sometimes. So what's your turnaround time on this?
the turnaround time is definitely not 1.5 to 2 years. It's rather straightforward because we'll be working with specific research communities to have these standards in place beforehand. So a particular assay, researchers will get together and decide, well, these are the variables which need to be reported and are the minimum gold standard for getting the certification. So that'll speed things up. And also in terms of revenue, the push is towards open publishing, and researchers have a small budget, $3,000 to $5,000, that they're spending on publishing in open journals. This will speed up the process, and it means they have the funds allocated to some fraction of that can be spent on getting our licensing.
there are already large entire papers, entire websites dedicated to various criteria for proteomic or RNA-seq or microarray. It goes on. There have been large conferences on all of these that already have this information out there. How do you propose to actually look into people's individual experiments and see how compliant they are? Isn't this too complicated to turn into kind of a cookie cutter look and see whether they're compliant or not?
I see it as an ideal middle ground. So I'm a cancer metabolism researcher myself, and I use a lot of websites like GeneCards and PhosphoSite as a reference, which parses the data and makes it easy for me to figure out what I want immediately. But the other extreme is me spending all this time familiarizing myself with an assay and becoming an expert in it. So this middle ground here will require either machine learning or researchers coming together through some forum and deciding on, okay, here's one particular assay, a Western blot. Please report the membrane type, that's a must. Please report the particular voltage machine used, that's a must. And then these can be used for someone in the laboratory starting a project to make sure they use papers that meet this middle ground criteria, and they neither need to become an expert nor do they have to start from scratch.
I don't know. I have to say I don't understand exactly what you're proposing. The number of variables that would affect success or failure in any series of experiments is so high that it's very difficult for me to imagine that you're going to actually be able to add value this way. What you just said was actually speaking specifically to, say, the type of membrane that you use for a Western blot. Generally speaking, those types of issues are fairly well understood, and groups use relatively the same types of equipment or materials. I think the variables that you'd have to tackle in this kind of situation are, the animal model that's being used, whether it truly represents the disease, the actual biochemistry and biology that you're trying to examine, the cell lines, are they grown the same? The level of intricacy that you'd have to get to, in my opinion, would be extreme.
To some extent, that's true. I feel like a lot of the issues you're raising about the animal model and whatnot are questions that are at the discretion of the reviewer of the article. But
well, again, all articles go through an extensive review process before they're published. Whether Amgen or another company can actually repeat the experiment, the number of variables that dictate whether they can repeat an experiment or not are quite significant. I don't know how this is going to work.
But
Okay.
when we say repeat, we don't mean we have to reproduce, we don't have to experimentally do the experiment, but you have to allow us to be able to do it.
let me give okay.